We're live.
Is anyone even on the?
I haven't like.
For those on the Zoom, good morning. This is the California Institute for Regenerative Medicine. The meeting of the Application Review Subcommittee will be starting late today in about 10 minutes at 9:30. Good morning, board members and members of the public who are rolling in. Just to repeat our earlier announcement, the board meeting of the Application Review Subcommittee will be getting started in about 5 minutes. Thank you. Good morning, everyone. We're just waiting for one or two more members. We're just about to get started. Just hold tight a few more seconds. Good morning, everyone. This is Scott at the CIRM headquarters. Believe we have a quorum now, ready for the Application Review Subcommittee meeting. Vito, if you would like to kick things off. You are muted, Vito.
Thank you. We can start with the roll call, please.
Give me a second. Okay. Right. Dan Burnell. Maria Bonneville. Judy Chow. Leandra Clark Harvey.
Present.
Anne Marie Dulege. Isabel Duron. Mark Fisher Colbury.
Here.
Fred Fisher.
Here.
Elena Flowers.
Present.
David Higgins.
Here.
Vito Imbasciani.
Here.
Stephen Julesgaard.
Present.
Rich Lahara.
Present.
Christine Miaskowski.
Present.
Lauren Miller-Rogan.
Here.
Adriana Padilla.
Present.
Joe Panetta. Marvin Southard.
Here.
Carol Watson. Carol, I thought I saw you.
Sorry, I was having trouble unmuting. I'm here.
No worries. Kevin Zhu. All right. We are good.
I was having problems unmuting as well.
Oh, Dan?
Yeah,
Okay, great. Thanks, Dan.
thank you.
We can proceed.
Vito, you're muted.
Again. Sorry, that was accidental. We can start now. I don't have my agenda in front of me, but I think at this point Gil takes over.
That's right. Gil, are you ready to go?
Yes, I will be sharing a presentation. And today I have with me Dr. Haley Lamb. So she is the Associate Director of the Review Office, and she's going to help me go through the 6 different applications that we have for consideration today. And these 6 applications, just as we begin, cover actually 2 different cycles of May and June of the Grants Working Group. So that's just something to take note of. As always, we start with our mission, which is to accelerate world-class science to deliver transformative regenerative medicine treatments in an equitable manner to a diverse California and world. Our budget for this fiscal year, which starts in July, we're just at the beginning of this. So we had an allocation that was approved by the board of $252 million. The amount requested today in total from the 6 applications is about $50 million, leaving, if approved, about $200 million balance for the remainder of the fiscal year. The scientific scoring system that's used to grade the applications that come into the clinical program use a system of 1, 2, or 3. A score of 1 means that an application has exceptional merit and warrants funding. And that's the case for all the applications that you're going to be seeing today. Some applications will receive a score of 2. Those typically go back to the applicant to address concerns from the review panel and will resubmit within a short time. And then those that receive a score of 3 means those are sufficiently flawed that they don't warrant funding, and those can't be resubmitted for 6 months. The scientific review criteria that the grants working group uses in order to come up with a score is based on the following 5 questions. First, does the project hold the necessary significance and potential for impact, meaning what value does it offer? Is this something that is worth doing? Does it have a good rationale? Is it well planned and designed? And is it feasible, including having the appropriate qualified individuals and all the resources to carry out the activities that are proposed? And then finally, does the project uphold the principles of diversity, equity, and inclusion in the project? That last component, the DEI, is both considered by the scientific members, as just mentioned, under the 5th review criterion, but it is also considered separately by our board members, patient advocate members that serve on the grants working group through a separate DEI score. We provide a rubric, which is shown in the image that we don't expect you to read, but just so you are aware that that rubric exists and it helps guide the scoring for our board members. And the scoring system for DEI is based on a scale of 0 to 10, with 10 being the most outstanding response or score. The composition of the grants working group itself includes several roles. There are the scientific grants working group members, of which there are 15, and they provide scientific evaluation. And we put together panels that include diverse expertise, including disease area expertise, regulatory, CMC, product development, and so on. And so they provide the scientific score that you see. As also mentioned, we have our grants working group board members who are patient advocate or nurse members of the board. They conduct the DEI evaluation, provide a patient perspective on the significance and impact of these projects, and provide oversight on the overall review process itself. And then lastly, we have scientific specialists who are non-voting members. So they participate in an ad hoc basis whenever we need additional expertise. They provide a scientific evaluation, but they don't provide a final scientific score. All right, so the first application that we're going to consider today, and then just to note, Haley and I are going to go back and forth a little bit, given the order of the applications were arranged based on our ability to have all members participating and voting on these. So the first one that we're going to consider is CLIN 2-14068. This one is entitled Treatment of Severe Aplastic Anemia by Induction of Mixed Chimerism using CD4 positive T-cell depleted haploidentical donor stem cell transplant. So this therapy is a donor blood stem cell transplant that has its T-cells depleted in order to avoid graft versus host disease, and it's combined with a low toxic conditioning regimen. And the indication is for patients with severe aplastic anemia, and their goal is to complete a phase 1 first-in-human clinical trial. The funds requested is just over $9 million. No co-funding is required for this applicant. Just some background on the severe aplastic anemia. This is a life-threatening disease of the immune system in which the blood cells, blood lymphocytes specifically, destroy other blood cells, resulting in severe anemia and bone marrow damage. It is, in effect, an autoimmune disease. The condition can be cured with a bone marrow transplant, but the approach is less effective in older patients, typically over 40, who experience a higher incidence of the graft failure or graft versus host disease. And additionally, patients that don't have a fully matched donor, which often is the case for many underserved groups, are associated with inferior survival outcomes. So the proposed therapy offers the potential for improved outcomes by significantly decreasing the occurrence of GVHD and eliminating the need for a fully matched donor. And why is this a stem cell or gene therapy project? This involves a stem cell transplant. Currently, we don't have anything in our active awards portfolio that addresses severe aplastic anemia. So this would add a new project to that portfolio. In terms of previous funding by the applicant team, there is a related project by one of the team members, not the same PI, but the team essentially is largely the same. This was a CLIN 2-stage project that was focused on sickle cell disease. The project outcome was a phase 1 clinical trial. They had several milestones. The project was not completed in part because of the COVID pandemic that severely impacted patient recruitment. And I believe subsequently the PI has retired, so that project has not continued. The Grants Working Group recommendation for this application is as follows. There were 11 members that gave this a score of 1, 2 that gave it a score of 2. The DEI score is 8, and the CERN team recommends funding in concurrence with the Grants Working Group recommendation for the amount of just over $9 million. So Dr. Bassiani, it's back to you for any discussion or questions.
Thank you. I'd like to open it up first for comments from the board members. Thank you, Gil.
Vito, this is Scott. It might be appropriate to ask if there is a motion on the table to fund the application.
Oh, that's right. Thank you, Scott. Okay, so I need a motion and a second, of course, to launch discussion.
Moved, Marvin.
Marvin moves. Do I hear a second?
I'll second.
That was Steve Julesgaard. Thank you. Okay, discussion first from the board. Mariana, I can only see a few board members at a time.
There are none. There are no hands. Oh, wait, hold on. Yep, there are no hands raised at the moment.
Okay. Are there any members of the public attending the meeting that would like to comment?
If there are, press *9 to get put in the queue, and then *6 to unmute. I do not see any popping up.
All right. Then, and Scott, could you please take a roll call vote?
Sure. So this is to fund application 14068.
Yes.
Dan Bernal,
Aye.
Mark Fisher Colbury,
Yes.
Fred Fisher,
Aye.
Elena Flowers,
Yes.
David Higgins,
Yes.
Vito Imbasciani,
Yes.
Steve Julesgaard,
Yes.
Rich Lahara,
Yes.
Christine Miaskowski,
Yes.
Lauren Miller-Rogan,
Yes.
thank you, Adriana Padilla,
Yes.
Marv Southern,
Yes.
Carol Watson,
Yes.
I'm sorry, Leandra, call your name, Leandra Clark-Harvey. And Leandra, are you muted?
She is muted.
Leandra, I'm sorry, we show you muted. And Leandra, one last time. Okay, that's fine. Veto, that is unanimous and the motion carries.
Okay, thank you very much. Gil, we can proceed with the second application.
Yes, so the second application has two recusals, Maria Bonneville and Steve Julesgaard, just to be aware. This application is CLIN 2-14748. The title is Evaluation of Safety and Feasibility of Cytomegalovirus-Specific Anti-HIV Chimeric Antigen Receptor T cells in people with HIV. So the therapy itself is a CMV specific T cell that expresses a chimeric antigen receptor, so it's a CAR T, and it targets HIV infected cells. The indication is for people living with HIV/AIDS. The goal is completion of a phase 1 first-in-human clinical trial. The funds requested is 11.3 million, and no co-funding is required for this specific phase and applicant type. Some background information. HIV, as we know, severely weakens the immune system, and infected individuals become susceptible to other infections and some types of cancer. And antiretroviral therapy, or ART, is a lifelong treatment that is used to manage HIV infection, but it is not a cure, and it is often associated with high morbidity and costs. And so more effective potential curative treatments are needed. People living with HIV maintain a pretty high level of T cells that are specific to the cytomegalovirus, and this is a very common infection. By the time one reaches the age of 40, about half of the population has been exposed to CMV. And so in people with HIV, T cells that are specific to this virus continue to be quite functional. And so the proposed approach leverages that fact to create a potentially curative CAR T cell therapy that can recognize and destroy HIV-infected cells and then remain actively vigilant by way of that CMV signaling. Why is this a stem cell or gene therapy project? The therapeutic candidate contains blood progenitor cells and involves gene manipulation. Other projects in our portfolio that are similar, we have 3 other clinical stage projects that are for HIV/AIDS as an indication, in one case focused on AIDS lymphoma. These candidates, among the 3, are very different approaches. One is a gene therapy, the other is a CAR T cell that's similar to this project. And then the last one utilizes genetically modified blood stem cells, where the goal is to replace the immune system with HIV-resistant cells. So one project has a similar approach, but does not use the CMV element as a component to try to make the therapy perpetual, as is done in this particular project that we are considering today. The applicant has had a long history of funding from CERN. So I included in this table what could fit, but there is one additional CERN award beyond that. The applicant has had a good history in terms of performance, with milestones that are proposed having been appropriately accomplished, some with some minor delays, and one that was specifically delayed because of COVID-19 pandemic, but otherwise generally very good performance on all CERN awards. The Grants Working Group recommendation for this project is a score of 1, with 12 members giving it a score of 1. There was 1 member who gave it a score of 2. Nobody gave it a score of 3. The DEI score was a very high score of 10. And the CERN team recommendation is to fund this project for the award amount of 11.3 million.
Well,
Dr. Bassian.
Yeah, thank you, Gil. Great presentation. Looks like a great application. Open now to, I need a motion and a second to.
So moved. So moved, Bernal.
Bernal.
Second,
Thank you, Dan. Who seconded?
Fred
Fred
Fisher.
Fisher.
Fred Fisher. Great, thank you. So board members? Comments?
Do you see?
There are none currently.
There are none. This investigator must have must go back very far in time to have that much experience with CERM. A great track record. So no comments from the board members. Is there anyone responding from the public, Mary Ann?
I'm checking. And for the public, if you do have a comment, *9. And I don't see any.
Okay. Then we can proceed to a vote on the application, number 14748. Scott, can you take the roll?
Sure. Dan Bernal,
Aye.
Leandra Clark Harvey, Mark Fisher Colbury,
Yes.
thank you. Fred Fisher,
Aye.
Elena Flowers,
Yes.
David Higgins,
Yes.
Vito Imbassiani,
Yes.
Rich Lahara,
Yes.
Chris Miaskowski,
Yes.
Lauren Miller Rogan,
Yes.
Adriana Padilla,
Yes.
Marv Southard,
Yes.
Carol Watson,
Yes.
thank you, and I'll call Leandra Clark Harvey. Just a minute, just doing a little math.
Mhm.
Great, that's 12 ayes and no noes, so the motion carries.
Well, thank you very much, Scott. Gil, we can now proceed to the third of the 6 applications.
Okay, Haley's going to present this next one.
Okay.
Good morning. So this application for discussion is CLIN 2-14787. And the title of this project is a Phase 2B randomized assessor mass clinical trial to assess the safety and efficacy of a retinal pigmented epithelial implant in subjects with geographic atrophy. And the product itself is a patch with a layer of stem cell derived retinal pigmented epithelial cells on a matrix. And the indication this product is aiming to treat is geographic atrophy, which is a late stage form of age-related macular degeneration, AMD. The goal of this project is the completion of a Phase 2B clinical trial. Funds requested are 12.37 million and co-funding is provided of 8.25 million and 40% co-funding is required for this category of trial and applicant. So a little bit of background information on this, on AMD. So it's a leading cause of vision loss in the developed world. And as mentioned before, the geographic atrophy is a late stage form of AMD, of dry AMD, where the supportive cells, these retinal pigment epithelial cells, degenerate over time. And this degeneration of these cells over time contributes to the death of the photoreceptors that they support within the retina and then leads to visual impairment and sometimes blindness in the late stages of disease. So the proposed therapy here would be potentially a scalable approach to replace the disease portion of the eye that is damaged and promote survival and function of those supportive RPE cells and thus protect the eye from disease progression and vision loss. And in some cases, potentially, in fact, improving vision with this treatment. So this project is a combination therapy with a supportive matrix that also contains stem cell derived RPE. Current CERM portfolio projects that are active, there's one translational stage. This is preclinical and another that is a CLIN 1 preclinical stage. And both are slightly earlier stage than this one, which would be a clinical trial. Prior applicant funding from CIRM is related to some of the key personnel that are also on this project who have received funding for the development of this project, essentially through 2 disease team awards for the earlier stage IND enabling and a phase 1 study of the same product. So finally, the GWG recommendation for this application is to recommend for funding with 9 votes as a score of 1, 5 votes as a score of 2, and no votes for a score of 3, and a DEI score of 9.5. The CIRM team concurs with this recommendation, and for the same award mentioned earlier, $12.4 million.
Thanks very much for the presentation. May I have a motion, please, from the board to commence discussion?
So moved, Marvin.
Marvin moves.
Second.
Second, Mark Fisher-Colbert.
Thank you, Mark. I have a second. So comments from the board on this proposal, 14787?
There are no hands raised.
No hands are raised. We are all stunned by the brilliance of the scientific community. Anyone from the public would care to comment?
Adriana raised her hand.
Great.
I just wanted to ask the team, can they discuss why there was 5 on a score of 2, just for our review?
Yes, I could just briefly mention that there were some questions mainly around, so there was maybe 2 things. One was that there was the technique for the implantation itself, they thought might be challenging. So there was some concerns that maybe there would be some variability across the different trial sites. It wasn't, I don't think any of these concerns were sort of major concerns, but things that were brought up by the reviewers. And then the other concern by one of the panel members was regarding the placebo group, which it wasn't clear from the statistics that were proposed by the group on how the placebo group would be integrated into the analysis of the total patients or participants within the trial itself, and how that would feed into potentially a phase 3 trial. So I don't think there were any concerns that necessarily couldn't be adjusted if it came back as a tier 2. So
I think one other thing to add is to remember that a score of 2, typically reviewers give a score of 2 for where they want clarification or additional information. So it doesn't necessarily indicate that they would not want the project funded, but rather that they're looking for additional information typically.
And was that given to the reviewers?
So no, I think reviewers will typically come down on scoring, between a 1 or a 2. So if they score a 2, they will provide comments where they feel they would have liked additional information or clarification. So to the points that Haley raised, but the majority felt that the project was fine as it is, that it didn't need to come back to the grants working group. So when questions may be left open like that, in terms of having some of the grants working group members having a question, we usually will follow up with them. If the
That's what I wanted to know.
Yeah, so we will follow up as part of the process prior to funding to make sure at least those are points that are considered.
Perfect, thank you.
Mm-hmm.
Any other comments from the board members? If not, Mary Ann, just take a look for the public comment.
Okay, Vito, we do have one in the room. You have 3 minutes for your comment.
Great. Yes, I'd like this is Jane Lipkowski. I'm actually the PI and applicant on this application. Just wanted to clarify a couple of questions that were brought up about the difficulty of the surgery. In the phase 1/2A clinical trial, we successfully implanted 15 different patients. And now we're looking in this next clinical trial, one of the major objectives of the trial going forward is to look at having more surgeons implant to establish that, in fact, this procedure can, in fact, be established across a wide group of retinal surgeons. We don't believe that that will there will be a problem, but that is part of the objective of this particular grant. The second question was about the placebo group. We're sorry, it appears that maybe one of the reviewers had didn't understand the role of the placebo group. But we are going to compare both on safety and efficacy, compare the results from the treatment group versus the placebo group. I think one other question that was brought up during the review was, what is the role of a natural history group? We are going to, in this program, look at also comparing the data from a very broad range of clinical trials where the natural history of or sham controlled groups in other people's clinical trials. We will also compare data from our trial to those of control groups in other clinical studies.
Great. Thank you.
Thank you, Professor, for your explanatory comments. Are there any other comments from the public?
And a reminder, *9 if you do want to be placed in the queue. And I see none, Vito.
Okay. Well, thank you very much, everyone, for your comments. Discussion is closed. Scott, we can proceed to a roll call vote on this application.
Yes, and again, just to restate, the motion is to fund application 14787. Dan
Correct.
Bernal,
Aye.
Leandra Clark Harvey, Anne Marie Deliege, Mark Fisher Colbury,
Aye.
Fred Fisher,
Aye.
Elena Flowers,
Yes.
David Higgins,
Yes.
Vito Imbasciani,
Yes.
Yes.
Steve Julesgaard,
Yes.
Rich Lahara,
Yes.
Chris Miskowski,
Yes.
Lauren Miller Rogan,
Yes.
Adriana Padilla,
Yes.
Martin Southard. Thank you. Carol Watson,
Yes.
Maria Bonneville.
Yes.
That worked. Thank you, Maria.
Thank you.
2, 3, 4, 5, 6, 7, 8, 9, 10, 14. And the motion carries 14-0.
Wonderful. Great. So Gil's team, whoever's going to present the next one, the fourth application.
That will be me. Yeah. So the next application is CLIN 215087. Title of this application is Phase 1 Study of Chimeric Antigen Receptor Engineered T cells targeting CD33 for the treatment of relapsed refractory acute myeloid leukemia. This treatment is immune T cells from a patient's transplant donor engineered for targeted leukemia killing. And the indication is relapsed refractory AML. The goal is the completion of a phase 1 clinical trial, and they are requesting just under 12 million with no co-funding required for this application. Some background information, the clinical background. So relapsed refractory AML is a type of leukemia that's either returned after prior treatment or hasn't responded to prior treatment. And available treatments right now are not very effective, and this type of leukemia is known to have poor outcomes. So the proposed therapy, given the therapeutic options are limited, their new options for patients are needed, and the proposed therapy uses a targeted approach to kill the cancer cells and hopefully lead to improved outcomes for patients. This is a gene-modified cell therapy. And similar CERM portfolio projects that are active right now. There is currently a CLIN2 phase 1 trial using a monoclonal antibody. And there is a CLIN1 IND preclinical project that is a vaccine candidate. And this particular applicant team has not received prior CERM funding. So the recommendation from the grants working group is to recommend funding with 13 votes in the scientific score of 1, 2 votes in scientific score of 2, and no votes for a score of 3. The DEI score received by this team was a 10, and the CERM team concurs with the recommendation to fund this application for just under 12 million.
Thank you very much for the presentation and the recommendation. May I entertain a motion to discuss?
Moved, Marvin.
Thank you, Marvin.
Second.
Did you get that, Mary Ann?
Sorry, who was
No.
the second?
Chris.
Adriana? I couldn't tell.
It was Chris
Chris.
Miaskowski. Thanks very much.
Okay, thank you. Thank you. Okay, the floor is open for discussion from board members.
No hands are raised, Vito.
Okay. Are you scanning for public comment?
I am. And reminder for public comment, *9. And I don't currently see any.
Okay. If there's any member of the public out there who struggled with making a comment, we could always come back at the end of the meeting. No, I guess we can after the vote is held. Sorry. Let's proceed to a roll call vote, Scott.
Great. And to restate the motion, it's to fund application 15087. Dan Bernal,
Aye.
Ray Bonneville,
Yes.
Mark Fisher Colbury.
Yeah.
Fred Fisher,
Yes.
Elena Flowers,
Yes.
David Higgins. And David, I'm not hearing you. Are you muted?
I think he's muted.
It's close. He's looking for it, I think.
David? No.
Yes. David, you're still muted.
Yeah, he's looking for it. I'll come back. Vito Amassiani,
Yes.
Steve Julesgaard,
Yes.
Rich Lahara,
Yes.
Chris Miaskowski,
Yes.
Lauren Miller Rogan,
Yes.
Adriana Padilla,
Yes.
Marvin Southern, Carol Watson,
Yes.
and David Higgins.
Yes.
Ah, very good. Thank you, David. And the motion carries 14-0.
Okay, thank you very much. So Gil, I'm going to turn it over again now for the 5th application for consideration.
Okay, so for this next application, just to note, Mark Fisher Colbury is recused. The application is CLIN1 14607. The title is Cancer Stem Cell Interception with a Small Molecule Splicing Inhibitor. So this therapy is a small molecule drug of splicing that selectively eradicates therapy-resistant cancer stem cells. The indication that is sought here to treat is secondary acute myeloid leukemia, or SAML, or high-risk myelofibrosis. The goal is to complete IND-enabling studies and file an IND with the FDA that will allow them to begin a clinical trial. Funds requested are 3.2 million. Co-funding is 800,000, which is 20% that's required under this project. So background on secondary acute myeloid leukemia This is a cancer that can develop from pre-existing high-risk myelofibrosis, which itself is a blood cancer that disrupts the body's normal production of blood cells. The 5-year survival rate for patients with secondary AML is only 26%, and standard of care therapies are generally not curative and can result in significant morbidity. A key factor for the severity and recurrence of many of these cancers and other blood cancers as well is the persistence of cancer stem cells of the tumor. So in terms of the value proposition, this proposed therapy blocks a key RNA modifying enzyme that tends to promote cancer stem cell persistence. And as a small molecule drug, the approach, if successful, could provide a very practical and feasible option for patients with this disease. Why is this a stem cell or gene therapy? The candidate is a small molecule that acts on cancer stem cells. In terms of other projects in our portfolio of active programs, we have a CLIN2 that's addressing AML and CMML, that's a monoclonal antibody, and a CLIN1 that's a vaccine for AML. These are the same projects that Haley mentioned regarding the other AML project. In terms of previous CIRM funding for the applicant team, we have another person who has also had a long history of CIRM funding and might even outdo the previous one with 3 projects that are not on this slide. But in all cases, these projects, which have varied from infrastructure programs such as the Alpha Clinics, Translational Disease Team, and early stage candidate discovery, have been all completed on time with few, if any, delays in the project. So they have a very good track record with previous CIRM funding. The grants working group recommendation for this project is to fund with a score of 1, and it received a unanimous score by 15 members. There were no scores as a 2 or a 3. The DEI score is a score of 9, and the CIRM team recommends to fund in concurrence with the grants working group recommendation for the amount of 3.2 million. Dr. Imbasiani.
Yes. Thank you, Gil. I'm going to open the floor to a motion to consider discussion 14607.
So moved.
Thank you, Fred.
I'm happy to second.
Good. Thank you, Anne-Marie.
And hello to everybody, by the way.
Hello. Okay, the floor is open to discussion. Was there a hand, Emery?
No.
No hands raised.
Oh, Marianne, I'm sorry, I had it backwards.
No hands raised.
Okay,
Vita.
no hands are raised. Any comments from the public?
Star 9 from the public, if you want to make a comment. And I don't see any.
Okay, that being the case, Scott, we can proceed to a roll call vote on CLIN1 14607.
The motion is to fund 14607. Dan Bernal,
Aye.
Maria Bonneville,
Yes.
Fred Fisher,
Yes.
Elena Flowers,
Yes.
David Higgins,
Yes.
Ido Imbasiani,
Yes.
Steve Julesgaard,
Yes.
Rich Lahara,
Yes.
Chris Miaskowski,
Yes.
Lauren Miller Rogan,
Yes.
Adriana Padilla,
Yes.
Marv Souther,
Yes.
Carol Watson,
Yes.
Anne Marie DeLiege,
Yes.
Great, the motion carries 14-0.
Motion carries. Thank you very much. Okay, Gil or Haley, I don't know who's got the last presentation.
Yep, that's me. So just a note that there is a conflict with one member of the ARS, Fred Fisher, and to move on to the application itself. And I believe this is the last one. So Claim 14933. The title of this application is the manufacturing of an antisense oligonucleotides for a phase 1/2 clinical trial for amyotrophic lateral sclerosis. And the therapy is an antisense oligonucleotide, and the indication is ALS. And the goal of this is to complete the manufacturing activities and file an IND with the FDA. The funds requested are just under 2.2 million, and with a 20% co-funding that's required of just over $500,000, and the maximum funds for this category is 4 million. So a little bit of background information. ALS is a neurodegenerative disease that results in the death of nerve cells in the brain and spinal cord, causing the muscles in the body to gradually weaken and eventually leading to the loss of limb function, difficulty breathing, paralysis, and eventually death. So there are medications that can slow down the progression, but no cure for this disease. Proposed product would offer an opportunity to treat ALS patients with various underlying causes, and potentially have a greater impact on patient quality of life than the current standard of care. And the therapeutic candidate is a gene therapy. So current CERN portfolio projects, there is a CLIN2 phase 1 clinical trial running right now for the same indication, and this is a different approach with genetically engineered stem cells. The applicant does not have prior CERN funding. And finally, the scientific recommendation from the grants working group was a 13 recommendation to fund with 13 votes for tier 1, 1 vote for tier 2, and none for tier 3, with a DEI score of 7. And the CERN team recommendation is to fund the application for the amount of 2.2 million. Dr. Bassiani?
Yes, great. Thank you. So for this final application, could we have a motion to discuss?
Can make the motion, yeah, to discuss.
Yes, Marie moves and
Second, Marvin.
Marvin seconds. Thank you. Comments now from board members.
There are no hands raised, Peter. Oh, hold on. There's Adriana.
Yes, can the team just summarize what were some of the issues regarding the DEI score of 7, please?
So I think the main concerns were that it didn't seem like there was a lot of early stage engagement, DEI engagement activities to be conducted under this specific award. And it wasn't clear from the explanation of the applicant how this therapy would potentially impact underserved and disproportionately affected communities.
Does the team go back and ask the applicant to address those issues?
And so I think that the CERN team can work with the awardee if funded to improve those DEI plans. It's part of it's just part of the ward administration here at CERN.
Okay, thank you.
Adrienne, thank you. Those are the two questions that we're hoping would be asked. Any other comments from board members?
Just maybe a clarification on my part, and I apologize because I haven't had a chance to really look at the most recent exchange of emails in great detail, but is there any issue here? What I see is unanimity, at least from a scientific perspective, if I'm correct, as well as the CERN team concurs with the WG. So do I miss something is my question.
Gil?
No, not that,
Okay. Thank you.
I can tell.
Thank you.
Mm-hmm.
Scanning once more for the public. Star 9.
There are none.
Okay, there are none. Scott, we can proceed then to a vote on this final application.
Right, and the motion is to fund application 14933. Dan Bernal,
Aye.
Maria Bonneville,
Yes.
Anne Marie DeLiege,
Yes.
Mark Fisher Colbury, Elena Flowers,
Yes.
David Higgins,
Yes.
Vito Imbasciani,
Yes.
Steve Julesgaard,
Yes.
Rich Lahara,
Yes.
Chris Miaskowski,
Yes.
Lauren Miller Rogan,
Yes.
Adriana Padilla,
Yes.
Marvin Southard,
Yes.
Carol Watson.
Yes.
Thank you very much. The motion carries 14-0.
Well, good. Thank you, Scott. Just an editorial comment from the chair. I want to compliment the applicants on the quality of their applications submitted to this session of the board, to judge by the near unanimity of the people doing the evaluations. And with the exception of the sixth application, the high scores in the DEA category. I don't have a long institutional memory, and I think this question is rhetorical, but I wonder if we've ever had a meeting with so many high-scoring DEAs presented at the same time. I'm sure Scott or Gil will tell me that answer later. Thank you, everyone. Is there any other business for the board members? Scott?
No, there isn't. That concludes the business of the ARS, unless there is any other public comment regarding an item not on the agenda.
Mm-hmm.
There are none.
Doesn't
There
appear.
are none. Okay, can we adjourn, or do we need a motion?
You can adjourn.
We can adjourn. Thank you, everyone. This was a very, very productive meeting, under an hour. I appreciate your participation. Thank you.
Thank you.